Hi! We are the Procard Unit!
In this video, we are going to discuss the Chapter 9, Host Response to Infection. But first, let me just give a background on how this topic applies in our daily living. The human body's ability to respond to infection is a complex process, integral to our survival against pathogens. Kaya kapag yung mga microorganisms like bacteria, viruses, fungi, or parasites invades to our body, the host or our body sets up a series of defenses to contain and get rid of those microorganisms. This defense system, known as the host response to infection, and it involves many parts working together, and that will be discussed thoroughly later on. As we tackle this topic, we will be encountering terms that are involved or essential for us to understand this chapter. First, we have antigen, a substance recognized by the immune system, whether by the B cell or the T cell, that serves as the target of the immune response but may not necessarily lead to an immune response. Next, we have epitope, the structure in the antigen that is recognized by the B cell or the T cell. And then, hapten, a substance that is of low molecular weight that can only induce an immune response if bound to another substance that is already immunogenic, or the carrier molecule. Next, we have immunogen, any substance capable of inducing an immune response, whether humoral or cell-mediated or both. And lastly, we have immunology, the study of the immune system and the immune response. Now, let's proceed to the properties of antigens. Simply, an antigen is a foreign substance that enters our body, and it triggers our body to produce antibodies. And antibodies, on the other hand, is a protein produced by our immune system to attack and fight off these antigens. There are several properties that an antigen must possess to make it immunogenic. And basically, immunogenic refers to the ability of a substance to trigger an immune response in the body. When something is immunogenic, it can activate the immune system to produce antibodies or initiate other defense mechanisms to fight off a potential threat. The first property is the foreignness and genetic composition. The immune system recognizes and responds to substances that are considered foreign, meaning they are not part of the body's own cells or tissues. An antigen must be different enough from the body's natural structures for the immune system to see it as something that needs to be attacked. Genetic composition refers to how unique and the antigen's genetic makeup is compared to that of the host. Second is the chemical composition and complexity. Antigens with a more complex chemical structure are more likely to be immunogenic. This is because the immune system can recognize complex patterns and structures more easily. Proteins and large carbohydrates, which have diverse and intricate structures, are generally more immunogenic than simpler molecules like lipids. Next is the molecular size and stability. Larger molecules are typically more immunogenic than smaller ones because they have more sites for the immune system to recognize. Additionally, antigens need to be stable enough to remain intact long enough for the immune system to detect and to respond to them. If an antigen breaks down too quickly, it might not be recognized. And lastly, the mode of entry of the antigen. How an antigen enters the body can affect its immunogenicity. Antigens that enter through the skin, respiratory system, or gastrointestinal tract might trigger different types of immune responses. The route of entry can also influence the level of immune response. Some modes of entry might lead to a stronger response due to the greater exposure to immune cells. Now that we already know the properties of an antigen to make it immunogenic, the question is, how does our body produces antibodies to fight these antigens? The immune system is composed of molecular and cellular components that are derived from the central or the primary and the peripheral or the secondary lymphoid organs. The central lymphoid organs are the primary sites for differentiation and maturation of the important cells that play an important role in adaptive immunity, which are the T lymphocytes or the T cells and the B lymphocytes or the B cells. This consists of the bone marrow and the thymus. The bone marrow is the site from where blood cells originate. The precursor cells of lymphocytes are found in the adult bone marrow, and this is where they differentiate into B cells and T cells. Once differentiated, B cells remain in the bone marrow and undergo maturation in the bone marrow. On the other hand, the T cells will go out of the bone marrow as immature and incompetent forms, the CD4 and the CD8. Then, migrate to the thymus where they mature and become competent. After maturation, the mature B cells and T cells enter the circulation through that fluid we call lymph and proceed to the peripheral lymphoid organs to await any antigen that may enter the body. The peripheral lymphoid organs consist of the lymph nodes, spleen, and the mucosa-associated lymphoid tissues, which include the tonsils, adenoids, Peyer's patches in the ileum, and the appendix. These organs are the site of reactivity of lymphoid cells. These are where antigens are trapped and subsequently encounter the T and the B cells. Antigens are brought to these peripheral lymphoid organs where the cells needed for their destruction are located. Both mature T cells and B cells are found in the peripheral lymphoid organs. So, that was just a brief background about the immune system. Now, let's dig deeper into the different kinds of immunity.
Next is the cells of the immune system. The various responses of the human system are mediated by specific cells and the substances they produce. These cells include white blood cells, the granulocytes, which contains the 50 to 80% of white blood cells, the lymphocytes, 20 to 45% of white blood cells, and the monocytes, which contains 3 to 8% of white blood cells. Neutrophils play a major role in acute inflammation as well as in bacterial infections. Lymphocytes and macrophages are mainly involved in chronic inflammation. The lymphocytes are the predominant inflammatory cells in viral infections, and macrophages are also predominant in chronic inflammation. In addition, cells that belong to the mononuclear phagocyte system play a crucial role as antigen-presenting cells. We also have the antigen-presenting cells. These are cells that are involved in the processing and presentation of antigens to the T cells. Follicular dendritic cells. Some of these cells are found in association with lymphoid follicles in the lymph nodes. Langerhans cells in the skin also brings antigens to the paracortical zone of the lymph node, where they are called interdigitating dendritic cells. These cells participate in the immune system by stimulating T lymphocytes and found in T cell areas of the peripheral lymphoid tissues. Dendritic cells are considered as the true link between the innate and the adaptive immunity. Eosinophils possess eosinophilic granules that play a role in type 1 hypersensitivity reaction, or what we call the allergy. It also secretes a substance called major basic protein, or the MBP, which is toxic to parasites, especially helminths or the worms. Like eosinophils, basophils also play a role in allergies. Granules of both eosinophils and basophils contain histamine, which when released is responsible for the changes seen during the initial phase of an allergic reaction. The platelets are membrane-bound cell fragments that are derived from large cells called the megakaryocytes. It is involved in blood coagulation. However, they secrete substances that play a role in inflammation. Natural killer cells are large granular lymphocytes that are also called the NK cells or the null cells. It is originally classified as cytotoxic T cells because they had the same manner of killing the target antigens. The figure 9.2 shows the origins of the cells of the immune system. The multipotential hematopoietic stem cell or the hemocytoblast differentiate into either the common myeloid progenitor or the common lymphoid progenitor. Common myeloid progenitor and the common lymphoid progenitor continue to differentiate along cell lines into lineage-restricted progenitor cells that become final and mature blood cells. Myeloid progenitor cells are precursors to the following types of the blood cells. And here we have the megakaryocyte, which contains the platelet, the erythrocyte, the mast cell, the myeloblast, which contains the basophil, the neutrophil, the eosinophil, and the monocyte. Under monocyte is also the macrophage. Lymphoid progenitor cells are precursors to other mature blood cell types. And here we can see the NK cell and the small lymphocyte. Under small lymphocyte is the T and the B lymphocyte, and under B lymphocyte is the plasma cell. We can see also the dendritic cell, which is present in the common myeloid progenitor and in the common lymphoid progenitor. So the next figure contains the white blood cells that play an important role in the body's immune responses. There are two types of white blood cell, the granular and the agranular. Granular are found in cytoplasm in the form of granules and has 4 lobes, while the agranular is a white blood cell without granules and only a single lobe. The granular contains the neutrophils, the eosinophils, and the basophils. Neutrophils is 60% of the total granulocytes, while the eosinophils is involved in destroying different parasites in the body. Lastly, the basophils are found in bone marrow or bloodstream, and it protects our bodies from different infections. On the other hand, the agranular contains the lymphocytes and the monocyte. The lymphocytes provide stability to the human body and prevent from serious infections, while the monocyte is the source of many other vital elements of the immune system, such as macrophages and dendritic cells. It also plays a role in both inflammatory and anti-inflammatory processes that take place during an immune response. T cells are located mainly in the paracortical and interfollicular areas of the lymph nodes and in the white pulp of the spleen, forming a thin sheet around the central arteriole or the periarteriolar lymphoid sheath or the PALS. They are involved in the body's cell-mediated immunity, and the T cells further differentiate into CD4 plus T cells and CD8 plus T cells or the cytotoxic or cytolytic and memory T cells. The CD4 T cells consist of the helper T cells, and the regulatory T cells contains the CD4 and the CD25 T cells. The helper T cells do not have the direct capacity to destroy an antigen. Instead, they activate the cytotoxic T cells and stimulates differentiation of B cells into antibody-producing plasma cells. The regulatory T cells play an important role in the maintenance of self-tolerance or the ability of the immune system to recognize self from non-self. They are the most abundant lymphocytes in the circulation, forming the body's immune surveillance, and the T cells, most especially the CD4 T cells, are the predominant lymphocytes in the circulation and constitute part of the body's immune surveillance. Some T cells also differentiate into memory T cells. Now let's proceed to the innate immunity. Innate immunity is also known as natural immunity. This immunity is already active from the time of birth prior to exposure to an antigen. Innate immunity is non-specific. It includes host barriers that prevent entry of microorganisms such as the skin and mucous membranes, which is the first line of defense. And the next one is the processes such as phagocytosis and inflammation. It is the second line of defense. It is activated within minutes following exposure to the antigen. The innate arm of the immune system performs two major functions. The first one is killing invading microorganisms, and the second one is activating adaptive human responses. The first lines of defense serve to prevent entry of the organism to the body and limit microbial survival. Physical and chemical barriers prevent attachment and entry of the organisms. These include the skin, fatty acids in sebaceous secretions, and sweat. Microorganisms that penetrate the first line of defense are prevented from multiplying inside the body by the body's second line of defense. If the primary barriers are breached, inflammation is activated as well as the natural killer cells. The microorganisms are recognized by innate immune cells and soluble mediators because of their molecular patterns called pathogen-associated molecular patterns or the PAM. Activation of inflammatory cells lead to phagocytosis of the antigen, while activation of the complement system results in the production of membrane attack complex or the MAC, which helps degrade the antigen. And lastly, the table 9.1 shows the important components of innate immunity, which contains the factors that limit entry of microorganisms and their mode of action, and the factors that limit growth of the microorganisms within the body. And lastly, the table 9.1 shows the important components of innate immunity, which contains the factors that limit entry of microorganisms and their mode of action, and the factors that limit growth of the microorganisms within the body and their mode of action.
Next, we have the adaptive immunity. Adaptive immunity is specific. It is activated after exposure to a particular antigen. Unlike innate immunity, it is an acquired response to an antigen that is initiated by recognition of specific epitopes or recognition sites of the foreign invaders. It involves production of antibodies by the B cells and activation of the cytotoxin T cells. Adaptive immunity response is delayed compared to innate immunity since it takes about 7 to 10 days. However, unlike innate immunity, the protection given by adaptive immunity is longer and, in most cases, lasts throughout the lifetime of an individual. Adaptive immunity possesses memory unlike sa innate immunity na hindi or it does not possess memory. The presence of these memory cells ensure a higher response once there is re-exposure to the same antigen, making the response amplifiable. Next, we have the immune response. There are two types of immune response, the primary and the secondary immune response. So first, we are going to tackle the first or the primary immune response. First exposure with an antigen leads to the activation of a specific set of helper T cells called the Th1 cells. Activation of these cells lead to activation of the inflammatory response and delayed type hypersensitivity as well as stimulation of B cells to produce IgM and IgG. The antibodies becomes detectable in the serum after about 7 to 10 days but can be longer depending on the nature of the antigen of the dose of the antigen. Next, I am going to discuss about the secondary immune response. Secondary immune response occurs after the exposure to the same antigen. A second encounter with the same antigen or a closely related one occurring months or years after the primary response will activate another set of helper T cells called the Th2 cells. Then, diba sa primary is it produced both IgM and IgG. Pag secondary immune response naman is lahat maliban sa IgM. Secondary response leads to a rapid antibodies na mas malala kaysa sa primary response. This explains by the persistence of antigen-specific memory cell. The predominant antibodies involved is IgG, and the levels tend to persist much longer than the primary response. However, if there is a need for other antibodies, some of the IgG in the circulation can undergo modification in their structure to become converted to another antibody. That process is called class switching. Lastly, we have the humoral immunity. Innate and adaptive immunity can be humoral or cell-mediated. Sa innate humoral immunity, it involves cytokines or cytokines and the complement system. Then, kapag adaptive humoral immunity naman, it involves the action of antibodies. Pag antibody-mediated immunity naman, it is directed primarily against extracellular pathogens, toxin-induced diseases, certain viral infections, and infections caused by encapsulated pathogens.
Antibodies. Antibodies are globulin proteins or immunoglobulins that react specifically with the antigens that stimulate their production. The most important function of antibodies are, first, to neutralize toxins and viruses, second, to opsonize microbes so that they will be readily recognized and more easily phagocyted. When we say opsonize, it is an immune process by which it uses opsonins to tag foreign pathogens for elimination by phagocytes. Third, to activate complement system. Complement system is the frontline defense for immune system which cleans up damaged cells, helps body heal after an injury or an infection. Fourth, to prevent the attachment of microbes to mucosal surfaces. Antibody structures. A typical immunoglobulin is shaped like letter Y and it consists of polypeptide chains linked by disulfide bonds. An antigen is a binding site which recognizes and binds to an antigen. Interchain disulfide bonds are found within each of the polypeptide chains. Chains are either kappa or lambda. They are produced by B cells during immunoglobulin synthesis. They are necessary to adjust BMN functions and assist pre-stimulation. Heavy chains protein are G, A, D, E, and M. Papain cleaves splits immunoglobulin within the upper part of hinge region, resulting into two Fab fragments and an intact Fc fragment. Complementary binding region is the main region engaged in antigen binding. Carbohydrates and glycoproteins play a central role in cellular invasion or infection by pathogens and the innate arm of the host of immune system. For variable loops, we have VL light chains, VH heavy chains, CL constant light chains, CH constant heavy chains. They are both responsible for binding to antigen and media tiny effector, function as opsonization and complement activation. There are 5 classes of immunoglobulins found in all species and all individuals. Each immunoglobulin class is defined by its component heavy chain. The first class, we have IgG. It is a monomer and the predominant antibody in the secondary immune response, or what we call as anamnestic response, and it is the major defense against bacteria and viruses. When we say anamnestic, it is renewed rapid production of an antibody on the second encounter within the same antigen. It is the most abundant antibody on the second encounter with the same antigen. IgM. It is the largest among immunoglobulins and is a pentamer. It has a J chain, or what we call as joining chain, and holds the IgM pentamer together. It is the main immunoglobulin produced early in the primary response, and it is the predominant antibody in an acute infections. Together with IgG, it can activate the complement system. It is more efficient activator of complement owing to its large size. IgA, it is called the secretory immunoglobulin, and it is the main immunoglobulin in secretions such as colostrum, saliva, tears, and as well as respiratory, gastrointestinal, and genitourinary tract secretions. It exists as a monomer in serum and is a dimer in secretions. Two monomeric units are held together by J chain. It is an important component of mucosal immunity, preventing binding of the organisms in the mucosal surfaces. Fourth class, we have IgE. It is called as reagenic antibody. It is medically important for two reasons. First reason, it mediates immediate and anaphylactic hypersensitivity reaction. The second reason, it provides defense against parasites such as helminths or worms. It binds to the surface of mast cells and basophils, where it serves an antigen receptor for allergen. It exists in monomeric form. For the last class, we have IgD. It is a monomer that has no antibody function. It is found on the surface of many B cells and serves as a surface marker for B cells, but may also function as an antigen receptor. It is present in all amounts of serum. Here are the comparison of the major classes of immunoglobulins. In conclusion, IgM is the primary response, fixes complement, monomer serves as a B receptor. IgG, main blood antibody, and it neutralizes toxins and opsonization. IgA is secreted into mucus, tears, and saliva. IgE, it is an antibody of allergy and anti-parasitic activity. And IgD is the B cell receptor.
Cell-mediated immunity refers to a branch of immune response that involves a specialized cell working together to combat infections and eradicate abnormal or infected cells. Cell-mediated immunity stands as cornerstone of the body defense mechanism against plethora or threats encompassing viruses, fungi, parasites, bacteria, and even tumor cells. In cell-mediated immunity has four basic functions. First is to provide resistance and aids in recovery from infections due to intracellular organisms. Example of this are viruses. Second is the important defense against fungi, parasites, and bacteria. Third, involved in transplant and graft rejection. 4. Main defense against tumor cells. The components of the cell-mediated immune system include several cell types such as the macrophages, natural killer cells, helper T cells, and cytotoxic T cells macrophages, together with the B cells and dendritic cells, present antigens to T cells. Cytotoxic T cell, also known as CD8 T cell, emerge as formidable warriors against intracellular pathogens and malignant cells, employing the perforin granzyme mechanism. CD8 T cell deliver a fatal blow to antigen. Upon antigen presentation, perforin punctures the membrane, enabling granzyme to induce apoptosis, effectively dismantling the trait. Next is the complement system. The complement system consists of a group of soluble proteins, C1 to C9, which are proteases that cleave and activate one another in a sequential manner. They are secreted as inactivate enzymes, which are enzymatically activated by another complement proteins. Next is the three-complement system. First is the lysis of cell. The complement system can directly attack and destroy pathogens by forming a membrane attack complex. This complex creates pores in the cell membrane of pathogens, causing them to swell and burst, ultimately leading to cell lysis. Second, the generation of inflammatory mediators. Complement activation results in the production of inflammatory mediators such as C3A and C5A. These molecules serve as potent chemoattractants and activators of immune cells, including neutrophils and macrophages. Third, opsonization leading to enhanced phagocytosis. This opsonized surface facilitates the recognition and engulfment of pathogens by phagocytic cells, such as macrophages and neutrophils. And it involves 4 basic steps. First, initiation. Second, formation of C3 convertase. Third, formation of C5 convertase. Fourth, formation of membrane attack complex or MAC. The alternative or properdin pathway is activated by bacterial products such as endotoxin or complexes of immunoglobulins. It is part of innate immunity and the first to be activated on initial exposure to the antigens. It bypasses a C1, C2, and C4 upon exposure to the antigen. The C3 component undergoes spontaneous hydrolysis and binds to activated factor B, forming C3BBB or C3 convertase. Next is the classical pathway of complement system is activated by antigen-antibodies complexes. These complexes binds to CLQRS or recognition unit. CLQ acts as the pathogen sensor. Binding of C1q to the antigen-antibody complex leads to activation of CLR, which activates C1s, leading to spontaneous activation and cleavage of complement proteins C2 and C4. Next is the mannose binding lectin or MBL pathway. Is activated by specific patterns of sugar found on the bacterial cell wall. Lectin, the first to be discovered, is the pattern recognition receptors that recognize the pattern mannose containing sugars. Number 1, C3 and C5, a chemotactic for neutrophils chemical mediator in inflammation causing vascular leakage or increased vascular permeability. Number 2, C3A, C4A, C5A function as anaphylactosins causing the granulation of mast cell and release of histamine. C5A is the most potent, followed by C3A, then C4A. Lastly, the membrane attack complex or MAC cause lysis of the bacterial.
Hypersensitivity reactions. Hypersensitivity reactions are exaggerated and inappropriate immune responses that lead to tissue injury resulting in a harm to the host. The injury may be brought about by the various substances and chemical mediators activated during inflammation, as well as the activation of the complement system. Hypersensitivity reactions may be categorized into 4 types: Type 1, Type 2, Type 3, and Type 4. So, Type 1 immediate hypersensitivity that are produced by the immune system in response to environmental allergens such as pollens, animal dunders, or dust mites. So, these antibodies IgE bind to the mast cells and basophils which contain histamine granules that are released in the reaction and cause inflammation. Type 1 immediate anaphylactic hypersensitivity, known as an allergic reaction and is mediated by IgE or immunoglobulin E. Type 1 hypersensitivity can be divided into 2 phases: immediate phase and late phase. So, immediate phase represents the vascular events of inflammation, which include vasodilation and increased vascular permeability.
major chemical mediator during this phase is histamine. Reactions occurs within minutes of re-exposure to the allergen and will eventually subside. 2 to 24 hours after the initial phase, there is recurrence of the symptoms of the patients. Late phase represents the cellular events of inflammation where the tissues will show infiltration by inflammatory cells which include neutrophils and eosinophils. Main chemical mediators during this phase include slow-reacting substance of anaphylaxis or SRSA, consists mainly of leukotrienes C4, D4, and E4. Prostaglandins also play a role during this phase.
Late phase. Also, late phase may develop 4 to 12 hours post early phase reactions and can last for up to 24 to 73 hours. And also, late phase reactions is mediated by eosinophils, neutrophils, and lymphocytes that can result in increased release of more mediates, which can lead to further tissue damage, swelling, increased erythema, cough, wheezing, and secretions.
The clinical manifestations are typical in a given individual. These are classified into local anaphylaxis and systemic anaphylaxis. So, local anaphylaxis include food allergy, urticaria, eczema, allergic rhinitis, or hay fever, and asthma. While systematic anaphylaxis is a served allergic response where patients manifest symptoms of circulatory collapse such as hypotension, severe bronchoconstrictions, and laryngeal edema.
First type, opsonization and phagocytosis. So, opsonization and phagocytosis in type 2 hypersensitivity reactions may occur when IgG antibodies bind to antigens on the cell's surface. Through its Fc receptors, so this antibody binding the cell for phagocytosis, drawing phagocytes like macrophages to engulf and then internally disassemble the cell. Skin prick test and scratch test. So, skin prick test and scratch test provides evidence for sensations and it can help to confirm the diagnosis of suspected type 1 allergy. And also, tests look for reaction to specific allergies that are placed on the skin.
Type 2 antibody-mediated hypersensitivity, known as cytotoxic or cytolytic hypersensitivity, involves three subtypes, two of which involves destructions of cells. A third subtype does not involve cytotoxicity, but instead involves cellular dysfunctions, which is the reason why the name was changed. This hypersensitivity reaction is mediated by IgG or IgM. The first subtype involves the process of opsonization and phagocytosis. Opsonization is the process where an antigen is coated by molecules that facilitate recognition by phagocytic cells, resulting in enhanced phagocytosis. Opsonizations of cell by IgG antibody can also cause activations of natural killer cells which can destroy the target cells without phagocytosis. This is known as antibody-dependent cellular cytotoxicity or ADCC. Example of conditions under the mechanisms are transfusion reactions, blood incompatibilities, autoimmune hemolytic anemias, and certain drug reaction that will lead to hemolysis or red blood cell.
First type, opsonization and phagocytosis. So, opsonization and phagocytosis in type 2 hypersensitivity reactions may occur when IgG antibodies bind to antigens on the cell's surface. Through its Fc receptors, so
This antibody binding the cell for phagocytosis, drawing phagocytes like macrophages to engulf and then internally disassemble the cell. The second subtype involves the process of complement and Fc receptor-mediated inflammation. This is initiated when antibodies IgG or IgM deposit in fixed tissues such as basement membrane or extracellular matrix. Acute rheumatic fever is an example of a condition under this mechanism. Second subtype, complement and receptor-mediated inflammations. So, by binding to self-antigens, antibodies can trigger the component pathway. The findings in the synthesis of complement components C3A and C5A, which functions of chemotactic factors for neutrophils, building them to the site and activating them. So, the tissues are damaged by the enzymes and reactive oxygen, species that these neutrophils subsequently release. Third subset, first example, myasthenia gravis. So, autoantibodies against the nicotinic acetylcholine receptors in myasthenia gravis prevent acetylcholine from binding to its receptors' muscle cells, which result in muscles weakness. First example, myasthenia gravis. So, autoantibodies against the nicotinic acetylcholine receptors in myasthenia gravis prevent acetylcholine from binding to its receptors' muscle cells, which result in muscles weakness. Myasthenia gravis is a neuromuscular disease that is characterized by progressive muscle paralysis. It is a neuromuscular disease that is characterized by a progressive muscle paralysis. The acetylcholine binds to the receptor found on muscle cells. Binding of the acetylcholine with its receptors will initiate muscle construction. In myasthenia gravis, autoantibodies against acetylcholine receptors binds to the receptor, preventing the binding of acetylcholine of the receptors. The muscles that are involved are initially those that are frequently used, such as eyelid muscles. The earliest manifestations of the condition is inability to open the eyelid, causing dropping of the eyelids or ptosis. Graves' disease. In this condition, an antibodies against receptor for thyroid stimulating hormone are produced. Thyroid-stimulating hormone normally stimulate the thyroid gland to produce thyroid hormone in cases when the hormone's level is reduced. The net result is continuous stimulation of the thyroid gland to produce hormones so that the patients manifest symptoms of hyperthyroidism. Graves' disease. So, in Graves' disease, the overproductions of thyroid hormones is caused by autoantibodies binding to thyrotropin receptors on thyroid follicular cells. Normally, blood levels of thyroid hormones control the pituitary productions of thyrotropin. However, these antibodies cause the follicular cells to produce thyroid hormones on their own. This process is unaffected by high blood levels of thyroid hormones, leading to much higher levels than what is necessary to cause symptoms hyperthyroidism.
Type 3 hypersensitivity reaction, also known as immune complex mediated hypersensitivity, occur when immune complexes form and deposit in virus tissues, triggering an inflammatory response. This can lead to tissue damage, especially in organs like kidneys and blood vessels. In this reaction, classical pathway of complement system is activated, leading to the recruitment of inflammatory cells and the initiation of inflammation at the site of immune complex deposition. This process not only destroys the target antigen but also damage the tissues where the immune complexes are deposited, causing fibrinoid necrosis, particularly in blood vessels. So, ano nga ba ang fibrinoid necrosis? Fibrinoid necrosis is a specific pattern of irreversible, uncontrolled cell death that occurs when antigen-antibody complexes are deposited in the walls of blood vessels along with fibrin. For example, in malignant hypertension, fibrinoid necrosis in the kidneys can lead to the kidney failure. And there are two forms of 3 hypersensitivity. First, local immune complex disease, and the second is the systematic immune complex disease. Local immune complex disease is exemplified by the Artus reaction, often seen as a complication of immunization with vaccines like DTP. It occurs when immune complexes precipitate in the walls of blood vessels at the injection site, leading to localized tissues necrosis. And next, systematic immune complex disease, on the other hand, is exemplified by acute serum sickness, triggered by administration of foreign serum, antibodies, or certain drugs like penicillin. Manifestations typically appear around 1 week after exposure and may include fever, urticaria, joint pains, lymphadenopathy, and splenomegaly. And next, we have the type 4 hypersensitivity reaction, formerly known as delayed type hypersensitivity, involve T lymphocytes rather than antibodies. This reaction results in tissues destruction either through inflammation caused by cytokines produced by CD4 T cells or through direct killing of target cells by cytotoxic CD8 T cells. A delayed type hypersensitivity involving CD4 T cells can be included by intracellular pathogens like Mycobacterium tuberculosis or certain viruses like fungi. For example, number 1, tuberculin skin test. Tuberculin skin test, this is a common diagnostic test for tuberculosis or TB. A small amount of purified protein derivative or PPD which contains antigen derived from Mycobacterium tuberculosis is injected under the skin. And next, contact dermatitis. Contact dermatitis is a type of skin inflammation that occurs when the skin comes into contact with an allergen or irritant. And next, rheumatoid arthritis. Rheumatoid arthritis is a chronic autoimmune disorder characterized by inflammation of the joints. CD8 T cells mediated cytotoxicity, on the other hand, leads to tissue destruction through the destruction of the cells bearing specific antigens on their surface. This mechanism is implicated in diseases like type 1 diabetes mellitus, graft rejection, and the destruction of viruses infected or tumor cells. Perforins and granzymes secreted by cytotoxic T cells play a key role in this process by inducing apoptosis of the target cells.
And now, allow me to present to you the figure 9.14 or the pathogenesis of type 4 hypersensitivity reaction, illustrating the mechanism of delayed type hypersensitivity involving helper T cells. And also the figure 9.15, the pathogenesis of type 4 hypersensitivity reaction, illustrating the mechanism of T cell mediated cytolysis. As a recall, Type 1, Type 2, and Type 3 hypersensitivity all involve antibodies, means they are humoral-mediated responses. Differ in hypersensitivity type 4 that does not involve antibodies, means it's not humoral-mediated response. It's a T-cell-mediated response. And now for us to really understand the figure 9.14, allow me to give you an example of this. One of the example of this, if we are exposed on poison ivy, a plant that produces toxic particles that may lead to rash and reaction on the skin. Poison ivy contains a compound known as urushiol that binds proteins in the skin and alter the protein, so that immune system now recognize the exchange skin protein as foreign. These immunogens are recognized the process by the antigen presenting cell and presented to a naive T helper cell. The APC will present a portion of the protein using an MNC class II receptor which T helper cell will able to recognize and bend to. After binding the APC to the T helper cell, it releases cytokines. It causes the naive T helper cell differentiate. If the APC releases certain cytokines including IL-12, then the naive T helper cell will differentiate into the type 1 plus helper cell known as Th7 cell. And after that, the cell will then release several cytokines including IFN gamma which activates macrophages. The activated macrophages will release IL-9 and TNF-alpha which causes expression of receptor on endothelial cells to recruit more leukocytes to the area. Activated macrophages perform phagocytosis and release reactive oxygen species and toxic lysosomal enzymes that cause the tissue damage that manifests itself after exposure to poison ivy. The APC may also release IL-6 and other cytokines that cause naive T helper cell to differentiate into a subtype of T helper cell known as Th17. Then Th17 cells then release IL-17 that activates neutrophils that then cause similar inflammatory actions as were caused by the macrophages. Remember that type 4 hypersensitivity is a cell-mediated and it takes more time to develop than other hypersensitivity types. This is because the T cells must be activated and recruited to cause the response as is known to occur after coming in contact with poison ivy. One can develop a rash known as vesicular dermatitis within 24 to 72 hours. For this reason, a type 4 hypersensitivity may also refer to as delayed type hypersensitivity. In CD8 plus T cell mediated cytotoxicity, the cytotoxic T cells destroy cells bearing specific antigens on their surface, leading to tissue destruction. This mechanism has been implicated in type 1 diabetes mellitus and plays an important role in the destruction of virus-infected cells as well as graft rejection and destruction of tumor cells. The principal mechanism of destruction involves perforins and granzymes, which are secreted by the cytotoxic T cells. Once the target cells are recognized by the cytotoxic T cells, they release perforin, which perforates the wall of the antigen. They also facilitate the release of granzymes, which in turn causes activation of caspases, thereby leading to apoptosis of the target cell. Now I will going to present to you the figure 9.15, the delayed type hypersensitivity. It is an immune response characterized dahil sa delayed onset inflammation and tissue damage upon exposure sa certain antigens. As you can see, this type of hypersensitivity is mediated by CD4 plus T cell, which is activated upon exposure to the antigen. Take note that CD4 plus T cell is the primary effector cells, while the CD8 plus T cell is generally plays a secondary role and both produce cytokines, and later on will lead to inflammation and tissue injury.
Vaccines. A great number of infectious diseases can be prevented by administering vaccines that induce either active or passive immunity. It contains a weakened, also called attenuated or inactivated form of the organism. It may contain the entire organism or a specific portion of the microbe, also known as subunit. It may also be derived from toxins produced by the microorganisms called toxoids. The immunization of a population stops the spread of infectious agent by reducing the number of susceptible hosts. Herd immunity. So in short, ang vaccine ay na-discover upang pasiglahin ang immune system ng ating katawan para mag-produce ng antibodies para ma-destroy kaagad ang mga organisms na papasok sa ating katawan. So there are two types of immunization, the passive immunization and the active immunization. In passive immunization involves the administration of purified antibody in preparation called immune globulins or antibody-containing serum. It is given for rapid, temporary protection, usually 3 to 4 months or treatment of a person. The protection given is short-lived. Immune serum globulin preparations are derived from infected humans or animals and are available as prophylaxis for several bacterial and viral diseases. Examples are human rabies, immune globulin, and immune globulins against hepatitis A or B, measles, chickenpox, and diphtheria. Immunization programs have achieved the following goals: protection of population groups from the development of common infectious disease such as pertussis, diphtheria, tetanus, and rabies. Control the spread of measles, mumps, and rubella, elimination of smallpox in the world. Passive immunization, it is used with the following goals of mind. First is to prevent disease after a known exposure. Second, to reduce symptoms of an ongoing disease. Third, to protect immunosuppressed patients. And fourth is to block the action of bacterial toxins and prevent the disease that they can cause. Active immunization involves the injection of vaccines prepared from organisms or their products. The response takes days to weeks to develop, but the protection given is long-term or even lifelong. Active immunization is preferred over passive immunization. So there are 4 major groups of vaccines. Number 1 is the live attenuated. Second is toxoid. The third is killed or inactivated, and the last is subunit. Vaccines are prepared using organisms with limited ability to cause disease and useful for protection against infections caused by development viruses. The first vaccine was developed by Edward Jenner for smallpox, and Albert and Sabin developed the first live oral polio vaccine. And there are 2 problems with the use of live vaccines. First, the organism may still revert to its original, virulent form once it enters the body. Second, the vaccine may be dangerous to immunocompromised patients and pregnant women. Immunization with a live attenuated vaccine resembles the natural infection leading to development of humoral, self-mediated, and memory responses. Immunity acquired is usually long-lived and, depending on the route of administration, can mimic the normal immune response to the infecting agent. Is toxoid vaccine. Toxoid vaccine were developed based on the principle that certain diseases are caused by exotoxins produced by the causative agent. Examples are tetanus, botulism, pertussis, diphtheria, and cholera. The toxoid were produced from the exotoxins. Toxoid vaccines are advantageous because they are safe without possibility of reverting to a virulent form. The component antigens are non-replicating, and they are more stable compared to live vaccines. The disadvantages include the need for adjuvant and multiple doses, and the possibility of developing a type 3 Artus reaction. Killed vaccines, in general, refer to vaccines derived from bacterial sources, while inactivated vaccines are derived from viruses. The first killed vaccine to be produced was the typhoid vaccine during the latter part of the 19th century. Examples of inactivated vaccines that are popularly used are the polio vaccine and the hepatitis A vaccine. Immune response to the killed or inactivated vaccines are similar response to toxoid vaccine, but in a wider range of antigen. Is the subunit vaccine. This type of vaccine is produced the same way as the killed or inactivated vaccine, but instead of using the entire organism as the antigen to stimulate antibody production, only a specific antigen or structure on the organism is used. The benefits of using subunit vaccines are similar to toxoid vaccine. An additional advantage is that with subunit vaccines, one can identify or distinguish the infected individuals from the vaccinated individuals. For example, in hepatitis B, immunized individuals will only have the antibody to surface antigen and will be the only ones detected in their blood, while infected patients will have additional antibodies present against the core antigen and the envelope of the hepatitis B virus. The disadvantages of using subunit vaccines are comparable to toxoid vaccines. Such vaccines also require multiple doses and addition of an adjuvant, as well as local reaction at the site of infection similar to the Artus reaction. Vaccines have been instrumental in the prevention of a great number of infectious diseases. However, the use of vaccines also entails certain problems. As mentioned earlier, live vaccines can occasionally revert to virulent forms and may be life-threatening when given to immunosuppressed individuals or pregnant women.